This lesson is about the practicals needed to show that chlorophyll, light and carbon dioxide are needed for photosynthesis. All of these experiments involve destarching a plant at the beginning. This means a plant needs to be put in the dark for 48 hours, so it uses up all its reserves of carbohydrates, so that any starch detected in the leaf starch test has come from the plant photosynthesizing during the experiment. Now, if you think back to the equation of photosynthesis, we said that carbon dioxide from the air combines with water from the xylem to make glucose, which is stored as starch. For the reactions to occur to produce glucose, we need energy from sunlight. This energy is absorbed by the chlorophyll in the leaf. The experiments you need to know are the experiments to show that carbon dioxide, light energy, and chlorophyll are needed for photosynthesis. Looking at chlorophyll, the need for chlorophyll was demonstrated in the previous lesson on the starch test of leaves. A variegated leaf was used. A variegated leaf has parts of the leaf that contain chlorophyll, the green parts, and parts of the leaf that do not contain chlorophyll, the white parts. When the starch test was carried out on the geranium variegated leaf, you can see that the central part that contained chlorophyll turned blue-black when iodine solution was added. This shows starch was present in the areas containing chlorophyll. The leaf stayed orange-brown in the areas containing no chlorophyll, showing no starch was present here. Therefore, photosynthesis only occurred in the areas of the leaf that contain chlorophyll, showing chlorophyll is needed for photosynthesis. To remind you, the starch test involves the following steps. First of all, boiling the leaf in water for 1 minute to kill the cells and disrupt the partially permeable membrane, so substances can pass in and out of the cell. Next, putting the leaf into a boiling tube. But first of all, you need to turn off the Bunsen burner so that we can pour ethanol over the leaf. Ethanol is highly flammable, so the Bunsen needs turning off. The leaf is then boiled in ethanol by putting the boiling tube containing the leaf and ethanol into the recently boiled water bath. Ethanol will boil as it has a lower boiling point than water. Boiling the leaf in ethanol removes the chlorophyll from the chloroplast in the leaf. Boil for 5 minutes. After 5 minutes, most of the chlorophyll will have been removed. You can see this as the ethanol has gone green. Take the leaf out of the boiling tube. It is now crispy, so wash it in the hot water in the water bath so it can be spread out on a white tile. Spread the leaf out on a white tile and add iodine solution. The white tile allows us to see the colour changes easily. The leaf will go blue-black where starch is present and stay orange where there is no starch. We now need to look at an experiment to show light is needed for photosynthesis. Therefore, we need to set up an experiment with the destarched leaf to allow light to get to some parts of the leaf but not to others. Here is a destarched geranium plant that my pupils have already used to set up their own experiments to show light is needed for photosynthesis. They have put their initials on strips of foil and then wrapped the foil around the leaf to block out light to areas of the leaf. The foil is pushed carefully around the leaf to block out any light. The plant will now be left in sunlight for several days so the plant can photosynthesize to produce glucose and store starch in the leaf cells. This is the result from the leaf that I set up. As you can see, it is orange around the outside and black in the middle. This shows that where the foil was, no starch is present, but where the light could get through, then starch is present, as shown by the blue-black of the iodine solution. Next is an experiment to show carbon dioxide is needed for photosynthesis. To carry out this experiment, we need to remove carbon dioxide from the air surrounding one of the plants, but have another plant as a control to compare it to. The control does get access to carbon dioxide. We set up the experiment by placing 2 geranium plants under bell jars. The bell jars mean they are isolated to the air just under the jars so we can control what gases are available to the plants. This plant is sealed with soda lime. Now, soda lime absorbs carbon dioxide from the air, so that means that this plant isn't going to get carbon dioxide from the air surrounding it. But we also have to have a control to prove that it's nothing else in this setup that is stopping the plant from photosynthesizing. So the only difference between the two is that this one has glass beads in a beaker and the other one has soda lime. Now, glass beads are not going to absorb carbon dioxide, so there's going to be carbon dioxide in the air around this plant. So this plant should be able to photosynthesize, but we will soon find out the results. So we'll leave these for a few days and then see what the results are by using the starch test on the leaf. So what have we learnt from this experiment? Well, if there's no carbon dioxide present, there's no carbon to make glucose, so photosynthesis cannot occur. If we remove the availability of light, then there is no light energy to convert to chemical energy, which is the whole role of photosynthesis. And if there's no chlorophyll, then there is nothing in the leaf to absorb the light energy needed for photosynthesis.